Welcome, dear students, to an engaging journey into the principles of crossing over, where you'll explore the fascinating cytological mechanisms that drive genetic diversity during meiosis. Together, we will uncover how crossing over not only drives genetic diversity, but also serves as the foundation for gene mapping, evolutionary biology, modern genomic research, and our broader understanding of life's architecture, covering the following learning objectives. We'll elucidate the cytological and molecular basis of crossing over in meiosis. We will examine the molecular processes that regulate and drive crossing over, evaluate the role of crossing over in producing genetic variation, and analyze the factors influencing crossing over frequency, master classical and modern gene mapping techniques. We will evaluate cytological and experimental proofs of crossing over and gene mapping to practical scenarios. Crossing over is the interchange of segments between non-sister chromatids of homologous chromosomes during the patchwork stage of meiotic prophase. It involves breakage and reunion of chromatids at precise points, leading to exchange of genetic material. This term crossing over was first used by Morgan and Cattell in 1912. Morgan's experiments with Drosophila melanogaster showed that genes are arranged linearly on chromosomes and may be inherited together due to linkage. However, recombination occurs through crossing over, explaining deviations from Mendel's law of independent assortment. The student of Morgan, namely Sturtevant, used recombination frequencies to construct the first genetic map in 1913, marking the beginning of chromosomal gene mapping. Bateson and Punnett in 1905 had noticed exceptions to independent assortment in sweet pea and introduced the concepts of coupling and repulsion. Today, molecular tools, cytogenetics and genomics have extended these classical principles, showing that crossing over not only generates genetic variation essential for evolution, but also underpins modern genetic mapping and breeding strategies. Now moving on to vital features of crossing over. Crossing over takes place during meiotic prophase, that is, during patchwork. Each pair of chromosome has 4 chromatids at the time. Crossing over occurs between non-sister chromatids. Thus, one chromatid from each of the 2 homologous chromosomes is involved in crossing over. It is universally accepted that crossing over takes place at 4-strand stage. Each crossing over involves only 2 of the 4 chromatids of 2 homologous chromosomes. However, double or multiple crossing over may involve all 4, 3 or 2 of the 4 chromatids, which is very rare. Crossing over leads to recombinations or new combinations between linked genes. Crossing over generally yields 2 recombinant types or crossing over types, and 2 parental types or non-crossover types. Crossing over generally leads to exchange of equal segments or genes, and recombination is always reciprocal. However, unequal crossing over has also been reported. The value of crossover or recombinance may vary from 0 to 50%. The frequency of recombinance can be worked out from the test cross progeny. It is expressed as the percentage ratio of recombinants to the total population. Moving on to cytological basis of crossing over. Students, crossing over is not random chaos, but it is a highly regulated biological process with a structural foundation. Cytological studies show how chromosomes align, pair, and physically exchange segments. Stages of meiotic prophase 1. Crossing over occurs during prophase 1 of meiosis, particularly in the patchwork stage. Liptoten. Chromosomes condense into thin thread-like structures, zygote. Homologous chromosomes undergo synapses pairing with each other via synaptonemal complex. Pachytene crossing over occurs at points called chiasmata. Enzymes such as SPO11 creates double-strand breaks. These breaks are repaired through mechanisms involving Holiday junctions, leading to exchange of genetic material. Diplotin, homologous chromosomes begin to separate and chiasmata becomes visible. Dikinesis, chromosomes condense further, chiasmata move towards chromosome ends, which is called terminalization. Synaptonemal complex. The synaptonemal complex is a proteinaceous zipper-like structure formed between homologous chromosomes during prophase 1 of meiosis. It plays a crucial role in synapses, ensuring precise alignment of homologous chromosomes along their length. The SC consists of lateral elements, central elements, and transverse filaments that stabilize the paired homologies. Its formation facilitates crossing over and recombination by holding non-cystic chromatids in close contact. The disassembly of synaptonemal complex occurs by diplotin, leaving homologous chromosomes connected at chiasmata. Molecular mechanism of crossing over. Double-strand break formation. Crossing over begins when the SPO11 enzyme deliberately introduces precise double-strand breaks in DNA of one chromatid during prophase 1 of meiosis. And unlike accidental DNA damage, these SPO11-mediated breaks are highly regulated, occurring at recombination hotspots to ensure crossing over at specific genomic regions, thereby promoting genetic diversity while preserving genome integrity. End resection. After the double-strand break, nucleases trim back the DNA ends to produce single-stranded 3' overhangs that are coated with specialized proteins for stabilization, and these exposed single-stranded regions act as the searching arms that locate and pair with complementary DNA sequence on the homologous chromosomes, enabling precise recombination. Strand invasion. The single-stranded DNA aided by recombinase proteins such as RAD51 and DMC1 invades the intact homologous chromosomes and displaces one strand to form a displacement loop or D-loop, marking the pivotal moment when genetic information aligns accurately between homologs to set the stage for segment exchange and increased genetic variation in gametes. Holiday junction formation. Once strand invasion is stabilized, the DNA strands from both homologs interlink to form a 4-stranded cross-shaped Holiday junction. That represents the physical connection between chromosomes, and this junction can migrate along the DNA to expand heteroduplex regions, thereby enhancing opportunities for precise recombination and error correction. Resolution. To complete recombination, specialized endonucleases cleave the Holiday junction in a manner that produces either crossover, that is exchange of chromosome arms, or non-crossover products, gene conversion without exchange. And this resolution step is vital for chromosome segregation, since crossover create chiasmata that hold homologs together until anaphase 1, ensuring accurate disjunction and reducing aneuploidy risk. Types of crossing over. Depending upon the number of chiasmata involved, crossing over may be of 3 types: single, double, and multiple as described. Single crossover. It refers to formation of a single chiasma between non-cystic chromatids of homologous chromosomes. Such crossover involves only 2 chromatids out of 4. Double crossover. It refers to formation of two chiasmata between non-sister chromatids of homologous chromosomes. Double crossovers may involve either two strands or three or all the four strands. The ratio of recombinants and parental types under these three situations are observed as 2:2, 3:1 and 4:0 respectively. Multiple crossover. Presence of more than two crossovers between non-sister chromatids of homologous chromosomes is referred as multiple crossing over. Frequency of such type of crossing over is extremely low. Moving on to cytological visualization of crossing over. Chiasma and crossing over. A chiasma is the visible point of exchange between non-sister chromatids of homologous chromosomes during meiotic prophase, first described by Janssens in 1909. Based on position, chiasmata are terminal at the ends or interstitial within the chromatids. The number of chiasmata per bivalent varies with chromatid length and may involve 2 to 4 chromatids. Chiasma terminalization. Terminalization refers to the movement of chiasmata towards chromosome ends between diplotin and metaphase 1. It has been explained by three hypotheses. Electrostatic hypothesis. According to this hypothesis, terminalization takes place due to localized repulsion force in centromere and generalized repulsion force on chromosome surface during diplotin stage. Coiling hypothesis. According to this hypothesis, terminalization takes place by mechanical tension developed within the chromosomes due to coils. Thus, tension force becomes greater than the force binding the chromatids at the point of exchange, resulting in terminalization. Moving on to elastic chromosome repulsion. According to this theory, all bodies having a definite shape resist any change that leads to alter their shapes. Chiasma forces the chromosome out of shape by its binding force. This leads to the development of repulsion at the point of exchange, resulting in terminalization of chiasma. Genetic recombination and linkage. Parental versus recombinant. Parental non-recombinant types. Offspring inherit the same allele combinations as parents. Recombinant types. Offspring show new combinations due to crossing over. Recombination frequency. Recombination frequency is calculated as number of recombinant offsprings by total number of offsprings into 100. 1% recombination is 1 map unit. RF less than 50% genes are linked. RF equals to 50% genes are sought independently, either unlinked or very far apart. Example, in a cross AB by AB into AB by AB, if 800 parental and 200 recombinant offsprings are produced, RF will be 200 divided by 1000 into 100. That is 20%. That means genes are 20 centimorgan apart. Gene mapping technique. Classical gene mapping techniques, 2-point test cross, involves two genes, test cross heterozygous AB by AB, with recessive small ab by small ab. Limitations. Double crossovers go undetected, leading to underestimation of distances. 3-point test cross involves three genes, involves the capital ABC by small abc into ABC by small abc. Double crossovers identify order. Middle gene has parental flanks in doubles. More accurate, double crossovers can be detected, allowing gene order determination. Multi-point crosses involves 4 or more genes used to create detailed genetic maps of entire chromosomes. Advanced gene mapping techniques. Molecular markers, restriction fragment length polymorphism, RFLP. uses restriction enzymes to detect DNA variation. Random amplified polymorphic DNA, RAPD, uses random primers to amplify DNA regions. Simple sequence repeats or microsatellites, SSR, repeated short DNA motifs used as high-resolution markers. Single nucleotide polymorphisms, SNPs, single base change is most abundant and widely used markers. Microsatellites, repeat variations for linkage. Fluorescent in situ hybridization. Fluorescent in situ hybridization techniques label DNA sequences, allowing visualization of gene locations and crossover events. Immunostaining of synaptonemal complex proteins. Modern cytogenetics uses antibodies against SC proteins to track recombination sites. Future directions. CRISPR-based mapping, target genome editing to introduce recombination sites. Single-cell sequencing, tracking crossovers in individual gametes. Students, let's move on to factors influencing crossing over. Frequency. Gene distance. Greater distance between genes increases crossover frequency. Closely linked genes show less than 1%. Interference. One crossover may suppress or enhance another nearby. Age. Crossing over decreases with maternal age, for example in humans. Drosophila. Sex. Higher in females, absent in Drosophila males. Temperature. Optimal 20 to 25 degrees Celsius. Extremes reduce frequency. Nutrition. Metallic ions, calcium, magnesium reduce recombination. Chemicals, mutagens, alkylating agents increase crossing over. Irradiation, X-rays, gamma rays enhance frequency. Chromosomal rearrangements, inversions, translocations suppress crossing over. Centromere and cytoplasmic effects, crossing over reduced near centromeres or by cytoplasmic genes, for example in pearl millet. Moving on to molecular mechanism of crossing over. There are two important theories for the possible explanation of molecular mechanism of crossing over. Copy choice theory. This theory was proposed by Belling in 1931. This theory states that the entire recombinant section or part arises from the newly synthesized section. The non-cystic chromatids, when come in close contact, they copy some section of each other, resulting in recombination. According to this theory, physical exchange of preformed chromatids does not take place. The non-cystic chromatids, when come together during pairing, copy part of each other. Thus, recombinant chromosome or chromatids have some alleles of one chromatids and some of the other. The information may be copied by one strand or both the strands. When only one strand copies, non-reciprocal recombination is produced. Drawbacks of the copy choice theory. According to this theory, breakage and reunion does not occur, while it has been observed cytological. Generally, crossing over takes place after DNA replication, but here it takes place at the same time. However, this theory is not completely satisfactory because there is no evidence of chromosome replication in two phases. The genetic material DNA has a continuous linear organization. DNA synthesis or replication occurs in interphase. A very small amount of DNA is synthesized during zygotine pachytene at the time of crossing over. Chromosomes at pachytene already consist of two sister chromatids, which were synthesized during the interphase. Moving on to breakage and reunion theory. Most accepted theory. Prior to crossing over, each chromosome of each bivalent get duplicated to form tetrad. This theory states that crossing over takes place due to breakage and reunion of non-cystic chromatids in pachytene stage. Crossing over involves the mechanical breaks in non-cystic chromatids due to twisting around each other, and reunion of chromatids takes place. According to this theory, first of all, chromatids break and then form chiasmata. Chiasmata results in crossing over. If the union takes place between sister chromatid parts, that is paternal to paternal and maternal to maternal, no genetic consequence is anticipated. However, if the break and reunion occurs between non-sister chromatids, that is parental to maternal or vice versa, recombinance would result. This hypothesis represents the best explanation to date to account for the formation of recombinance. Diagrams showing the stages involved in this hypothesis are given in. The first cytological evidence of crossing over was provided by Kurt Stern in 1931 in Drosophila. He used females with morphologically distinct X chromosomes, one broken X carrying the carnation eye color allele and dominant bar allele, and the other with a segment of Y attached, carrying alleles for normal red and oval eyes. When crossed with carnation males, females produced 4 gamete types, 2 parental, Car-B, plus, and 2 recombinant, Car plus and B plus. While males produced only parental types, since crossing over does not occur in Drosophila males. Cytological examination of recombinant progeny, red bar and carnation normal, showed exchange of chromosome segments between non-sister chromatids, proving that genetic recombination results from cytological crossing over. Similar confirmation came from Regeton and M.C. Klintock in maize. Significance of crossing over. It generates genetic variation, produces new allele combinations, ensuring diversity within population and providing raw materials for evolution. Ensures chromosome segregation. Chiasmata hold homologous chromosomes together, preventing non-disjunction during meiosis I. Facilitates gene mapping. Recombination frequencies help locate genes and construct linkage maps. Drives evolutionary changes. Variation created by recombination allows species to adapt to environmental pressures. Medical relevance. Abnormal crossover events may cause chromosomal disorders, deletion, duplication, translocation. Applied in breeding. Useful new gene combinations can be exploited in plant and animal breeding for improvement. Reveals chromosome structure, demonstrates linear arrangement of genes and aids in understanding gene action. Now, applications of gene mapping. Human genetics. Locating disease genes, for example, cystic fibrosis, Huntington disease. Mapping of cystic fibrosis gene on chromosome 7 used RFLP markers. Hemophilia patterns in royal families were explained through X-linked inheritance. Plant breeding. Developing molecular markers for selecting desirable traits. For example, disease resistance. High-density molecular maps in rice and maize accelerated the discovery of yield-enhancing and stress-resistance genes. Animal breeding. Identifying genes linked to milk yield, growth rate, or disease resistance. Chromosome organization studies. Understanding structural features of genomes. Evolutionary biology, comparing maps across species to study evolutionary relationships. Moving on to aberrant crossing over and chromosome disorders. Unequal crossing over leads to duplications or deletions, translocations, exchange of segments between non-homologous chromosomes may cause cancer or infertility. Invergence, reverse orientation of segments can suppress recombination. Dear students, crossing over is a central biological process ensuring genetic diversity, chromosomal stability, and evolutionary adaptability. Its cytological basis reveals the intricate structures and molecular mechanisms at play during meiosis. Gene mapping derived from recombination frequencies remains one of the most powerful approaches in genetics, now enhanced by molecular tools and genomic technologies. By understanding these principles, undergraduate students gain insights into how classical cytogenetics, molecular biology, and modern genomics converge to explain heredity, variation, and the architecture of life. We'll soon get back to you, students. Thank you.